Let's talk about preimplantation genetic testing in this video. This is the paper which was released in 2020 by Eshrey. It is based on preimplantation genetic testing, and they have divided the topic into various parts, and separately they have included PGT, M, SR, A. And before talking about different PGT, they have given an overview how PGT is carried out. How will we define PGT? It is defined as a test performed to analyze DNA from oocytes, because we can take the polar bodies or embryos which can be in the cleavage stage or blastocyst for either HLA typing or for determining the genetic abnormalities, which is the most common reason for doing the PGT, to determine the genetic abnormalities. But yes, it helps in selecting HLA matched child in some diseases, which the HLA matched child, if born, can help the sibling. So what is included in PGT? PGT A, PGT M and PGT SR. So this is the overview of IVF PGT process. So basic requirement of an IVF and PGT center is, firstly, we have to do the inclusion and exclusion criteria for each of the PGT M, SR and A. Counseling and informed choice is very important, because we might end up having no embryo which is suitable for the embryo transfer. So before undergoing the procedure, these things must be clear to the patient. Organization of preimplantation genetic testing. So proper counseling and informed choice is very important for any patient who is undergoing the preimplantation genetic testing. Obtaining the relevant documents, especially in the cases with PGT-M when it has to be performed, because the full maternal as well as paternal history, as well as in structural rearrangements when the balanced chromosomal translocations are there. So in such cases, the documentation becomes more important. Collecting the full family history becomes more important. The lab infrastructure, maintaining the equipment and the material, proper lab documentation, and regular accreditation and quality management. Coming on to the biopsy lab infrastructure, where the procedure has to be carried out. So for the areas within the PGT center, prior to each use, work surfaces and equipment should be cleaned and decontaminated with DNA decontamination solutions or 10% bleach, or by ultraviolet C irradiation or autoclaving. It is not recommended to use 70% ethanol. Why? Because it does not decontaminate the DNA. To prevent the contamination, protective clothing for DNA amplification of a single or few cells should be worn, including full surgical gown, hair cover, face mask, shoe cover. So all these things need to be worn, because this procedure requires a lot of precision, and since very few cells are involved, so DNA of the working personnel should not be transferred while taking the biopsy procedure. Gloves should be worn at all times and changed frequently. And what are the types of gloves which are important? These should be well-fitting, like nitrile gloves. But the vinyl gloves, which we usually use for the examination, those are not indicated. For areas within the IVF center, protective clothing, preferably with the low particle shedding, or non-powdered gloves and masks should be considered. The pre-amplification material and reagents should be kept away from any DNA source and preferably stored in a pre-amplification area. Now, what are the equipments and materials which are used for biopsy? First and foremost is the inverted microscope with heated stage. The second thing which is required is a three-dimensional micro manipulator and micro injectors, both air and oil, placed on anti-vibration pads. The third thing is a stereoscope for transferring oocyte or embryos in a biopsy dish and for tubing. Fourth one, obviously, the incubators, which can be box incubators, the time lapse, and the benchtop. Then lastly, a laser for assisted hatching and blastocyst biopsy. Coming on to insemination and culture. Firstly, the denudation is done in order to minimize the maternal cell contamination, because the oocytes will have the cumulus cells around them. So if we do not denude the oocyte, the possibility of cumulus cell which carries the maternal DNA might contaminate the overall result. So that's why denudation is done. ICSI is preferred rather than IVF because it also helps in minimizing the contamination. Following biopsy, the oocytes and embryos should be thoroughly rinsed to remove biopsy medium before culture or cryopreservation. Coming on to steps for biopsy. So first step is the creating an opening in the zona pellucida. Second step is removing either the polar body when we are taking the biopsy from the oocytes, or embryonic cells when we are taking the biopsy from the embryos. In case of the cleavage cell, it is the blastomeres, which is usually 1 or maximum 2, while in case of the blastocyst, it is the trophoectoderm cells, which will be anywhere between 5 to 10 cells. Zona pellucida opening. The opening of zona pellucida can be made by various methods. The most important method is the laser assisted drilling. But apart from laser assisted drilling, there are two more methods. One is the mechanical zona pellucida opening and another is the chemical zona drilling. We'll first discuss mechanical zona pellucida opening. It is also known as partial zona dissection. This method involves creating a slit in the zona pellucida using a sharp micropipette as shown in the image. The second one is the chemical zona drilling. It involves use of an acidic solution, that is the acid tyrodes, to locally dissolve the zona pellucida. As shown in the image, this is acid tyrode solution, so it helps in zona dissolution. Then third one is the laser zona drilling, which is the most commonly used method. This method involves use of guided non-contact laser beam, which can be adjusted to create a zona pellucida opening of the desired size in an accurate and rapid manner. This is the most popular method of zona pellucida opening. Now coming on to the sample removal, which can be polar body or embryonic cells. So there are 4 methods for the sample removal. First is the aspiration in the biopsy micropipette, simply aspirating the cells. Then second is the partial aspiration followed by pulling. Third one is the aspiration and excision with a laser. Fourth one is the aspiration and mechanical detachment. Aspiration in the biopsy micropipette is the most widely used method, and it is applicable for all stages of biopsy. Aspiration and mechanical detachment is also known as flicking. Coming on to sample collection. So once we have removed the sample, how to collect that sample and send it for the examination. First is the cell fixation. In case we do the cell fixation, further analysis by the fish analysis can only be done. Fluorescent in situ hybridization. Because Once the cells get fixed, the DNA amplification cannot be done. So it is the only fish method which can be used once the cells have been fixed. Second method is tubing. So the sample is put very precisely into the tube, and now this sample can be used for DNA amplification, and all the methods which involve amplification based testing can be used if the tubing has been done. So all these methods like quantitative PCR, next generation sequencing, array CGH, and SNPs. So all these methods can be used with the tubing, but with cell fixation only fish analysis can be done. Tubing is the most widely applied method for collection of biopsied samples. What exactly the tubing is? So we can see here in this image, the cell is shown, the blastomere, it is shown in the pipette which is above, and down we can see the tube in which the cell is going to be tubed. So the tubing has to be performed in a dedicated area and in a close proximity to biopsy area, because we cannot keep on shifting the collected sample here and there. So the tubing and the biopsy area, they should be together. They should be at close proximity. And wherever tubing is being done, that area should be DNA free environment. Now we will be discussing the biopsy stage and the procedure. So there are different stages where how the embryo grows, and at different stages we can take the biopsies. Firstly, before even the embryo has been formed, from the oocytes only, the polar body biopsy can be taken. We will be discussing more about the polar body biopsy in our next upcoming slides. On day 3, cleavage stage biopsy can be taken. And on day 5, 6 and 7, the blastocyst biopsy can be taken. So polar bodies are byproducts of female meiosis. It allows predicting the resulting genotype of maternal contribution to the embryo. It is crucial to distinguish polar body 1 from polar body 2. What is the time of biopsy? When should the polar body biopsy should be taken? So one is the simultaneous, that is both polar body 1 and polar body 2, both together we are taking. So what is the time for that? Polar body 1 and polar body 2 removed 6 to 9 hours after the insemination. We all know polar body 1 is already present when the oocyte comes out post retrieval, post oocyte pickup, while polar body 2 comes once the fertilization has happened. So 6 to 9 hours after the insemination, we will be taking the polar body 1 and polar body 2 if we have to take the biopsy simultaneously. Second is the sequential method. So in sequential method, the polar body 1 is taken within 4 hours following the oocyte retrieval. And polar body 2 following fertilization assessment. Usually the first fertilization check is done at around 18 hours. So 16 to 18 hours after insemination, the polar body 2 is taken. Procedure involved in polar body biopsy. The zona pellucida opening should be performed with laser or mechanically, and the diameter of the hole should be adapted to the diameter of the biopsy pipette. Although a second slit may be necessary to reach second polar body, but it should be avoided as it may affect the blastocyst hatching. Coming on to cleavage stage biopsy, it is done day 3 post insemination between 6 cell stage and the precompaction stage. So cleavage stage biopsy we can only do once the embryo has become at least 6 cell, and it must be done before the compaction happens, because once the compaction happens, that is the embryo becomes a morula. The cells, they get adhered to each other very tightly, and it becomes very difficult to take out the single blastomere. So before compaction only, we have to take the cleavage stage biopsy, which is mostly on day 3. It is recommended to biopsy embryos at 6 or more cell stage on day 3 with an acceptable grade. Fragmentation less than 25% is preferred. If fragmentation is more than that, the embryos might not be of good quality, and if the biopsy is taken, it might not be possible for embryo to grow further. So it is always preferred that the cell reaches at least 6 cell stage and the fragmentation remains less than 25%. So what is the procedure for cleavage stage biopsy? It is recommended to visualize the nucleus before taking out the blastomere to ensure that a nucleated cell is removed and to avoid binucleated cell for fish. Biopsied blastomeres are then fixed or tubed for further PGT analysis. Fixing and tubing we have already discussed in our previous slides. Coming on to blastocyst biopsy. Trophoectoderm biopsy at blastocyst stage enables removal of several cells for genetic testing while being non-invasive to inner cell mass, which is destined for the fetal development. So we all know it is the inner cell mass which becomes fetus and the trophoectoderm cells which becomes placenta. So whenever we are taking the blastocyst biopsy, we are not injuring the inner cell mass which is destined to become the fetus. So that is the advantage of blastocyst biopsy. What is the procedure involved with blastocyst biopsy? So blastocyst biopsy can be done in the 3 ways. Here in this image we can see the one with the clock and the calendar, that means days, and the one with only clock, that means hours. So the first method is day 3, the hatching is done. You can see here the cleavage stage embryo, the zona opening has been done. So wait for 2 days from day 3 to day 5. Once the embryo becomes day 5, it will automatically hatch because the zona drilling or laser zona hatching has already been done. So this is one method. And the morula based hatching is also one of the methods in which the day 4 the zona drilling has been done. Second method is same day hatching based method. So in this method, firstly on day 5 the zona drilling is being done, and wait for a few hours, the trophoectoderm cells will hatch out themselves. Then the third method is simultaneous zona pellucida opening and trophoectoderm cell retrieval, which might be a little difficult procedure because it is little difficult to pull out the cells when we do not give time for the cells to come out on their own. So these are the 3 methods which are being followed. Blastocyst biopsy procedure. ICM of blastocyst should be positioned between 7 and 11 o'clock so that it is clearly visible and distant from zona pellucida opening, as we can see from the image. Take care not to suction ICM by the holding pipette. The trophoectoderm cells are then aspirated into biopsy pipette with the gentle suction. We can see in the image, on the left side there is the holding pipette and on the right side there is the biopsy pipette. The cells are getting suctioned into the biopsy pipette. Laser pulses are directed at the junction between the cells to either excise the aspirated cells from the blastocyst or to minimize the cell damage while detaching the trophoectoderm cells mechanically. How many cells to biopsy? It is recommended to biopsy 5 to 10 trophoectoderm cells for genetic testing. The impact of removal of more than 10 trophoectoderm cells on embryo development remains an area of further investigation. So here in this slide, the 3 procedures have been compared. We can see the fragment origin, the polar bodies, which we all know they are waste products of maternal meiosis, while the blastomere or the cleavage stage biopsy cells are the totipotent cells, while the blastocyst or trophoectoderm biopsy, trophoectoderm gives origin to the placenta and extraembryonic membranes. So this is what the origin of the cells which we are going to biopsy. Number of cells retrieved in polar body, both are required, while in blastomere only one cell, two might be retrieved but it is discouraged. In blastocyst biopsy, 5 to 10 trophoectoderm cells. The meiotic errors assessment. In polar body biopsy, only maternal meiotic errors can be assessed, while in blastomere or cleavage stage biopsy, the meiotic errors, both paternal as well as maternal, can be assessed. The same goes with the blastocyst or trophoectoderm biopsy, where both maternal as well as paternal can be assessed. Mitotic errors. Polar body biopsy cannot tell about the mitotic errors, while the blastomere biopsy also cannot tell about the mitotic errors. While the trophoectoderm biopsy, it is possible within given technical, methodological and biological limitations. So still it is possible. The inconclusive diagnosis in polar body biopsy and the cleavage stage biopsy is higher, approximately 10%, while it is less than 5% with the trophoectoderm biopsy, obviously because we are taking more number of cells. What are the alternative biopsy methods? The first alternative biopsy method is the morula stage biopsy, and the second are the less invasive or non-invasive biopsy methods. The less invasive is blastocentesis and non-invasive is spent culture media. Coming on first to the morula stage biopsy. The biopsy of morula stage embryos on day 4 is performed after artificial decompaction. I already told in the last slide that because the morula, the cells are highly adherent to each other, so it becomes difficult to take out a single cell. But if we have to take the morula stage biopsy, firstly we have to artificially decompact the cells. It is characterized by loss of intercellular contacts and re-establishment of a spherical cellular shape. It is technically similar to cleavage stage biopsy but allows procurement of same number of cells as blastocyst biopsy. So if we are able to take the morula stage biopsy, the number of cells will be more, but technically it is more difficult than the cleavage stage biopsy. This technique requires more evidence before the broad clinical implementation. Then alternative biopsy methods. First is the blastocentesis. Blastocele fluid contains cell-free genomic DNA. The DNA can be purified and amplified for downstream genetic testing. The efficacy and accuracy of this technique are insufficient and need further elaboration before being clinically applicable. Coming on to the spent culture media, which is the least invasive method. Cell-free genomic DNA obtained from embryo culture medium may be potentially used for genetic testing. One of the limitations of technique is current inability to discriminate embryonic DNA from other sources of DNA contamination. And further optimization of the methodology is required. So thank you friends. This was all about the basics of the pre-implantation genetic testing. So we will cover the topics of PGT M, SR and A separately in our upcoming videos. Thank you. Thank you all.